Hello friends, Ace Botane, next episode. In this episode, we'll be discussing molecular basis of inheritance, 9th one. Normally one more needed. So in that one, we'll be trying to finish Human Genome Project and fingerprinting. So we can proceed. I will share the screen so it will be convenient for you. I think screen came, so we can proceed. So topics for discussion in this molecular basis of inheritance, 9th one. As usual, there will be a small introduction. Then the main aspect of this episode, regulation of gene expression, Operon concept. Then, as usual, 5 theory question, 5 NEET model question with the key. So as an introduction, so we know DNA is the genetic material connected with an idea called gene. So when a gene should be active, when it will be inactive, how this mechanism works. So regulation of gene expression and related matter connected with the gene working will be discussed in this episode. So idea you got what all things we'll be discussing in this episode. So we can enter regulation of gene expression. Idea is simple. All genes are not always working. Some genes may be working continuously throughout, while some genes not at all working, never working. Some genes will be work at some time. So when the genes will work. So this idea is basically coming under a term called differential gene action. How it is regulated, who will control it. So all this aspect comes under regulation of gene expression. See, gene expression can be regulated in several levels. In eukaryotes, regulation is exerted at 4 levels. That is NCERT usual question. And which are or what are these 4 levels? One is transcriptional level. Other is processing level that we already done in previous episode. That is regulation, splicing, all this, intron exon. Third one, transport of mRNA from nucleus to cytoplasm. And fourth one, connected with the protein synthesis, translational level. So that is how gene expression can be regulated in eukaryote at 4 levels. Important question for theory entrance, NEET model question, NCERT, page number 99. Now, approximately 50 to 90% of the primary transcribed RNA, mRNA, is discarded during processing. That already we studied, splicing, etc. The embryo, the development and differentiation into adult is also by coordinated regulation of several sets of genes. It is linked with metabolism, physiology, environmental conditions, all this. So coming under or together work under regulation of gene action. So many factors. Typical example is, suppose if you remember or if you can remember the terms like programmed cell death, apoptosis, where Nobel Prize was given some years back, then differential reproduction, a Darwinian concept you may be studied in evolution, etc. So all these are connected with the genes and the involved procedure. I think this first page is clear. So we can proceed to second page. In eukaryote, we discuss 4 levels, but in prokaryote, control of rate of transcriptional initiation is the predominant site for the control of gene expression. Basically, that fact that no separate nucleus for prokaryotes. So predominant site for control of gene expression is prokaryote, control of rate of transcriptional initiation. Again, connected with the NCERT important question. Gene products, that is regulatory proteins, can act in a positive manner that is often called activators, or in negative manner that is often called repressors. Again, NCERT page number 100, all related to NCERT. Now comes the famous idea called Operon concept. Scientist photograph is given there, name also given, Francois Jacob, Jacques Monod. Jacob is a biologist, otherwise genetist, and Monod is a biochemist. The Operon concept proposed in 1961, around 1961. That is why I put a star there, connected with the 1961 year, and received Nobel Prize in 1965. So it is Nobel Prize winning work, regulation of gene expression. Now, what is meant by the term Operon? That is a basic term, theory question. I'm not all involved in Operon, how the name came, NEET model question also. So an Operon means an assembly of genes, like a unit, an assembly. We study transcriptional unit, which includes structural genes, operator genes, promoter genes, and regulator genes. So all these genes will work in a coordinated manner, connected with specific metabolic pathways. So that assembly is called operon with a specific aim. And many operons will be working. Typical example is lactose operon that we have to learn. Tryptophan operon, present NCERT not discussed. Arabinose operon, valine operon, histidine operon, all these are operon regulations. I think idea you got, that is what is meant by operon. We can proceed to next page. This is the typical NCERT diagram, very famous, simple diagram explaining everything regarding operon and their function, etc., genes, all this. Very prominent diagram for exam point of view, theory, NEET, everything. So first, we can analyze this diagram, genes, their function. This is a typical DNA. That is what they shown. It's a typical DNA. So when we start, first gene is called regulator gene, also called I gene. I put a star there with a green color so we can easily identify where is I gene or regulator gene. See, I gene will produce a protein called regulator protein. This regulator protein can work either as an activator, or that is in a positive manner, or as a repressor in a negative manner. Operator gene, that is here you can find operator gene, is controlled by this I gene product. That is why the term regulator. So I think that regulator gene, you got the idea what it is, where it is, all this. Second is promoter gene. Here you can find. See, promoter site, we already studied. This gene have the site for the attachment of RNA polymerase. Here you can find P. Here you can find P. Next is operator gene. Here you can find operator. This will receive the regulator gene product. And this gene will allow the functioning of the operon depending upon the situation, operator. Oh, then you can find structural genes. These are the structural genes. This will be involved in transcribing mRNA for polypeptide synthesis. And in structural genes, 3 genes will be there set by A that we will be discussing in page number 5. Next, we have to proceed with our original topic, lac operon. We can continue. See here, lac means lactose. Lactose operon worked in Escherichia coli. And in biomolecular study, lactose is a disaccharide made up of glucose and galactose. Lactose is also called milk sugar. This all you know, but as an introduction, just to mention it. I think this page is clear. We can proceed to next page. So when we start with this page, see, since it is Escherichia coli, it is prokaryote. So prokaryote means we already studied it is polycistronic mechanism. And this polycistronic structural gene is regulated by a common promoter P and a regulator gene. And lactose operon is a typical example for inducible operon. For us, only inducible operon, we have to learn according to present NCERT, repressible not there. Lactose can act as an inducer, can activate or turn on from usual turn off nature of the operon. So lactose, that is inducing from turned off to turned on. Lactose or allolactose can be inducer. Allolactose has beta-1,6 glycosidic linkage, while in lactose it is beta-1,4 glycosidic linkage between its galactose and glucose unit. I think up to that it is clear. Now, regulation of lac operon by repressor is referred to as negative regulation. Lac operon can be under control of positive regulation also, but NCERT clearly says it is beyond the scope of discussion at this level. Page number 101, it is mentioned. I think that idea is clear. Now, last aspect, in lac operon, there will be 3 structural genes, 1 regulator or inhibitor gene, I gene, 1 promoter gene, and 1 operator gene. This is assembly of operon. I think idea clear. Now we can proceed to how this system working. Again, pure NCERT diagram, easy. In a simple manner, we can proceed how it works. Meanwhile, structural gene, I told you, set by A. The function will be mentioned in page number 5. Here it is. LAC-Z gene codes for beta-galactosidase. For what? For hydrolyzing lactose to glucose and galactose. Each gene has got its own function. LAC-Y gene codes for permease, which will increase the permeability of the cell to beta-galactosidase. LAC-A gene codes for transacetylase, which will transfer acetyl group to beta-galactoside. And this transacetylase, you may be remembering in first year you studied in respiration, there is a reaction called link reaction where there is an enzyme called transacetylase under pyruvate dehydrogenase complex. So each gene function is important, and which chemical, what is the aim, and if any mutation takes place there, what will happen, etc. That part we'll be doing question-wise after finishing the theory. I think functions of three structural genes clear. Now the working. Working is so simple. I told you we are proceeding with the NCERT syllabus. These are highly complicated events, and even Nobel Prize was given to this work. So that much work means a depth we don't want here because NCERT we are proceeding. So we can proceed. Hygiene, how it works. See, in absence of inducer, that is, there is no sufficient lactose to break down it, how system will be working. Hygiene will be producing a protein called repressor protein. That is why here repressor protein. So in the absence of sufficient quantity of lactose, repressor will bind to the operator. So operator is practically we can say inactive now, simple term. So when operator is inactive, automatically we can find it cannot connect with the promoter gene. So promoter is practically seems to be inactive. Most simple manner I am telling. So promoter is inactive. That means from promoter, no message is sent to structural genes. So respective enzymes will not be produced. Here we can find. That is why repressor protein plus operator gene result is switched off condition. Switched off. I think that idea clear. That is what we need. Simple manner, if you explain this is the principle. Now it's countdown. How it will be switched on. See, again, I gene or R gene will produce a protein, repressor protein. Here you can find. This repressor will be combining with the inducer. Inducer is lactose or even allolactose. Here we can say lactose. So inducer, lactose, and repressor protein will combine. So that means repressor become inactive. So repressor protein combines with the inducer producing inactive repressor. That is why this combination they given here, inactive repressor. So inactive repressor means practically we can say, easiest manner we can say operator gene is free. So operator gene free means it is sensed by promoter that will allow RNA polymerase to bind on P site. So from P site, message will be automatically proceeded to structural gene set by A. And they will be producing respective enzyme beta-galactosidase, beta-galactosidase permease, beta-galactosidase transacetylase. So it is switched on. That is why repressor protein plus lactose, inducer switched on. This is how the negative control works. Here it is switched on. It's a simple diagram given by NCERT, easy to follow. I think idea you got. Now we can proceed to next page. Okay. See, there are two systems of operon action. One is called inducible operon, other is called repressible operon. Inducible operon, example is lactose operon. Repressible operon means stop, repressed. Example is tryptophan, not in present syllabus. When it was there, detailed study was there, how many structural genes, etc. In lac operon, only 3 structural genes, etc. So we can leave tryptophan operon. Remember, a very low level of expression of lac operon has to be present in cell always. Otherwise, lactose cannot enter into a cell, permease enzyme, etc. That is given in NCERT page number 102. Now comes two terms not directly given but reference in NCERT. Constitutive genes and non-constitutive genes connected with the page number 101. So constitutive gene. So the term constitutive means basic. Constitutive means basic, fundamental, starting onwards, constantly, etc. Thus, constitutive genes means constantly expressed in a cell because their action and their products are highly needed to normal cellular activities always. So you may wonder, such genes are there? Yes. Example, in lack Operon, the role of I gene, inhibitor gene. Energy-related enzymes like ATPase and genes, energy should be always there. Respiratory genes like glycolysis and related genes. So these all are continuously working. So that is called constitutive gene. I think idea clear, even though NCERT not directly telling about the terms constitutive and non-constitutive genes, but connected with the page number 101. There is a reference in, I think, in NCERT first paragraph, there is a reference. That is why I put these two terms. Non-constitutive genes. These genes are also called luxury gene because it will not be working always, luxury. Since they are not always expressed in a cell, these genes will work according to the need of the cell and its activities. Example is in E. coli, lactose system, that is structural genes, ZYA genes. I think idea clear to all of you. So constitutive and non-constitutive gene. After that, 5 theory question you can find here and 5 NEET model question with the key. So what all things we learned in this episode, that is molecular basis, 9th one. We can proceed. First, we have an introduction. And we discussed there what all things we'll be learning there. Then regulation of gene expression. When a gene will be active, inactive, differential, gene action, etc. Then how it is regulated in eukaryote 4 levels. Then what are the 4 levels? Then we discussed about program of cell death, differential reproduction, etc. Then we entered into second page where that idea of Operon concept we developed. Before it, prokaryote, where is the principal control? That is, prokaryote control of rate of transcription initiation is the predominant site for the control of gene expression. Then we came into the photographs, scientist name, their profession, etc. And when they proposed and when they received Nobel Prize, all this. For all these years and the data, what NCERT given, we have to accept. Okay. Now, what is meant by an Operon assembly of genes? Then we entered into NCERT diagram. We can find. And each gene, regulator gene, what is their role? Positive, negative, activator, regulator, then repressor, etc. Then promoter gene, what is the role? P site, then operator gene, then structural genes, 3 genes. Details will be discussed later. Then we entered into lac or Operon or lactose. So Escherichia coli, disaccharide, monosaccharide, glucose and galactose, polycistronic, all this milk sugar. Then polycistronic structural genes here has got a common promoter and a regulator gene. Then lactose Operon is an example for inducible Operon. Lactose can act as inducer and usually that a turned off situation, making it to turn on. Then difference between lactose and allolactose. Allolactose is beta-1,6 glycosidic linkage, while lactose beta-1,4 glycosidic linkage. Then regulation of lac Operon normally under negative regulation can be positive also, but beyond our scope of discussion, page number 101. Then genes involved, that is 3 structural gene, 1 regulator or inhibitor gene, then 1 promoter gene, 1 operator gene, Operon, then how it works, that system. Initially, how it is switched off in the absence of inducer. So repressive protein attaches with operator, everything is off. That's the idea. Now 3 genes, ZYA, Z codes for beta-galactosidase for hydrolyzing lactose into glucose galactose. Then LAC-Y gene codes for permease, which will increase the permeability of the cell to beta-galactosidase. Then LAC-A gene codes for transacetylase, shifting acetyl group to beta-galactoside. And same transacetylase we studied in link reaction in after glycolysis respiration, going to Krebs cycle. Then how it will be on? In the equation we can find repressor protein plus operator gene switched off. Now switched on condition, that is repressor protein. Then inducer lactose is there, it has to break down. So repressor protein inducer will couple and it will be inactive repressor. So no connection to operator, operator is free. So sensed by promoter gene, P site, RNA polymerase bind, ZYA genes will be operating. Here you can find, here you can find, here inactive, here you can find it is activated and that will be producing respective enzymes. So you can find nutshell repressor protein plus lactose induces switch-on. Your NCERT diagram easy to follow. Then we discussed about two types of operon, inducible operon, repressible operon, inducible operon lactose, repressible tryptophan, not in our syllabus. And remember, a very low level of expression of lac operon has to be present in a cell always. Otherwise, lactose cannot enter into the cell NCERT page number 101. Now concerned with two more terms, constitutive and non-constitutive gene connected with the page number 101. I think first paragraph. Constitutive means continuously present. Example is, in lac operon, you can find the role of IG, energy-related enzymes like ATPase and genes. Similarly, respiratory process like glycolysis related, that should always work continuously. Non-constitutive gene, which is otherwise called luxury genes, that may not work continuously, but it is working sometimes. E. coli, lactose system, structural gene set by a gene. And after that, 5 theory question and 5 NEET model question with the key. Hoping that it is clear to you. If you like this all episode, subscribe all, like it, share it, comment on it. We can meet later with another episode. Thank you. Take care.